Hello.
Hello, sir.
Good afternoon, sir.
Hello. Good afternoon, sir.
Hello,
Good afternoon,
Jitendra, sir.
madam.
Hello, Jitendra, sir.
చెప్పండి మేడం.
సార్ హైదరాబాద్ నుంచి శ్రీనివాస్ మూర్తి గారే కదా సార్ ఇంట్రడక్టరీ రిమార్క్స్ చెప్పేది.
ప్రోగ్రామ్ షెడ్యూల్ ఇందులో షేర్ చేశాం కదండి.
యా 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 అంటే తను కన్ఫర్మ్ అవ్వలేదు అన్నాడు. ఓకే, అది కొంచెం కంగారులో ఉన్నాను.
లేదు మేడం.
yes sir.
మేడం షెడ్యూల్
Yes
కన్ఫర్మ్
sir,
అయింది
request
మేడం. As per the schedule,
రఘువీర్ వచ్చారు.
madam, I am there, madam.
రఘువీర్, ఇట్ ఇస్ శ్రీనివాస్ మూర్తి సరే కదా?
అవును మేడం, అవును మేడం.
ఫోన్ ఆన్ చేస్తాను, ఛార్జింగ్ కావాలి.
Madam, Purna madam, I think we should start the session. We'll be running late of time.
Yeah, shall we start? It's already 4.
Madam, yes, madam.
Yeah, we can start if all of them have joined. Means principals of all the colleges, if they have joined, we can start.
Okay, sir.
Sir, Srinivas Babu sir is
I
not
think
there.
Srinivas.
Srinivas Babu sir is on.
So anyone from pharmacy college, Vignan Pharmacy College?
I am there. My name is Manikiran.
Sir, Kiran garu. Could you find out about Srinivas Babu?
Yeah, our staff is also present over here.
No, Srinivas Babu garu. If sir comes, we can start.
Yeah, okay. We will let him inform. We have our staff and think they are following up. Pulipati Saujanya madam is also there. They are going to follow up.
Saujanya madam? So anyone from pharmacy college can respond so that we can start immediately. Sir?
Yeah, Dr. Jitendra, I'm trying to call him, but his mobile is out of coverage here, I think. I'm unable to reach him.
Okay.
I think I will try to with Alamanda. Okay, he's our technical person. I will try to call him. If there is any possibility, I will ask him to immediately report to principal. Okay.
Okay.
Yes, sir. Good evening. Students are waiting, sir. Can you please once again allow them? They're waiting in the waiting room.
Okay.
Devaraj garu. Devaraj garu.
Devaraj
Host. He is the host, sir.
Jalamanda.
Jitendra garu. Hello.
Sir, చెప్పండి sir.
Shall we start the program? Already 10 minutes go over now.
Okay, sir. We will start, sir.
Yes, we start.
So even speaker also has joined, sir.
Yeah, I know. He is the first person to join. As for our schedule, we are relating 10 minutes.
It's okay. Don't worry. Yeah. So for you, it is.
Okay, madam. Purna madam, you can start, madam.
Sir, Srinivas Babu, sir. Then
He will be joining.
I'll hand over. Then I'll hand over after my introduction to Srinivas Babu, sir. No, sir. If he doesn't get joined.
Direct మాట్లాడితే అనిపిస్తుంది.
Okay, sir. A very good evening to one and all.
Yes.
Warm welcome to Pharma webinar series. Respected Chair, Dr. Lau Rataya Garu, Vice Chairman of Vignan Group of Institutions, Sri Lau Sri Krishna Devaraya Garu, patrons, co-patrons, Principal of Vignan Pharmacy College, Badlapudi, Dr. P. Srinivas Babu Garu, Principal of Vignan Institute of Pharmaceutical Technology, Vizag, Dr. Y. Srinivasa Rao Garu, Principal of Vignan Institute of Pharmaceutical Sciences, Dr. M. Srinivasa Murthy Garu, and HOD Faculty of Pharmacy, VFST RU, Dr. C.H. Jitendra Garu. Today's speaker, Kakuman, dear fellow faculty members, students, and all the audience, a very good evening and warm welcome to you all. Myself, Dr. Purna Nagashri, working as an associate professor in Vignan Institute of Pharmaceutical Technology, Vizag. So this is a Pharma webinar series initiated by the pioneer and the visionary chairman, sir, Dr. Lau Rataya Garu, by combining all the 4 Vignan Pharmacy colleges from Guntur, Visakhapatnam, and Hyderabad, as well as from the university. During this pandemic season, this webinar series is started to inculcate and impart advanced knowledge and also to link the academic research with industrial research. So as a part of this Pharma webinar series, the first seminar is going to be given by Dr. Shrikant Kakumanu, sir, on overcoming challenges in the formulation of lipid-based nanoparticles and their present application. Now, I would like to invite the principals of the pharma colleges to give the brief introductory remarks. May I invite Dr. Y. Srinivasa Rao, sir, of Vignan Institute of Pharmaceutical Technology, Vizag, to give introductory remarks. Dr. Y. Srinivasa Rao, sir.
Good evening to Madam Purna Asri Garu. This is Dr. Y. Srinivasa Rao, Principal of Vignan Institute of Pharmaceutical Technology. This college was established in 2006 with the aim of giving scattering health needs to the society. Initially started with B Pharmacy. Later on, we started a M-Pharmacy with three specializations: pharmaceutics, pharma analysis, and pharmacology. In 2014-15, we started Pharma D program also. This is one of the large graduate in and around Uttarandhra area, and every year students, 95% students are passed out, and every year 85% of the students got placements. Remaining 15 to 20% the students they are going to higher education. This is the perspective of our Vignan Institute of Pharmaceutical Technology, and today it is fortunate we have guest Dr. Srikanth Kakumani Garu. He is expertised in lipid nano-based particles. So this is one of the interesting area, current scenario, and most of the drug delivery systems we are focused on hydrophilic drugs, and those drugs we are having hydrophobic nature. In particular issues, we are going to prepared lipid nanoparticles. So, in this, lipid nanoparticles are safe vehicles for drug delivery, made up of physiological related to lipids, and this nanoparticles already started last 20 years ago. Among the lipid nanoparticles have been engineered, the most important being solid nanoparticles, nanostructured lipid carriers, lipid drug conjugates, and lipid nano capsules. So in that case, mainly we are using the high pressure homogenization technique for the development. Right now, we are mainly focusing new technologies currently emerging for the lipid nanoparticle preparations, and these are easy to scale up. Even some process parameters can negatively influence the stability over time, and these can be administered by various routes according to the different therapeutic target. But as per the webinar topic, we are overcoming the challenges in the formulation of lipid-based nanoparticles and their present applications. Right now, everybody knows about the cancer and the drugs reach to the blood-brain barrier and the protein and the gene delivery. These are the main emerging topics nowadays for lipid nanoparticles application. This is the brief summary about the lipid nanoparticles. We are fortunate to have such type of wonderful research scientist and academician of Srikanth Kakumani Garu. He definitely will may give some inputs, and after completion of this webinar, we will may also focusing on the lipid nanoparticles, which is useful to our society. Thank you, madam, for giving this opportunity.
Thank you, sir. Thank you for your remarkable words. Now, may I invite the principal of Vignan Institute of Pharmaceutical Sciences, Hyderabad, Dr. M. Srinivasamurthy, sir, to give his brief introductory remarks.
Okay.
Dr. M. Srinivasamurthy, sir.
Respected speaker of today's webinar, Dr. K. Srikanth Garu, my friends, Dr. Srinivas Bab Garu, Dr. Y. Srinivas Rao Garu, Dr. Jitendra Garu, coordinators of 4 pharmacy institutions, faculty members of all the 4 pharmacy institutions, students and participants of other institutions, a very good evening to one and all. And on the behalf of Vignan Institute of Pharmaceutical Sciences, Hyderabad, I welcome you all for the today's webinar titled Overcoming the Challenges in the Formulation of Lipid-Based Nanoparticles and Their Present Applications. I express my sincere thanks to our beloved chairman, sir, Dr. Lau Ratai, sir, for bringing all the 4 pharmacy institutions on the common platform and initiating this webinar series. I strongly believe that one has to work individually as well as a team member. And I congratulate all the principals of other 3 pharmacy colleges for applying this principle among the staff members. I also congratulate Dr. Jitendra Garu for starting the first webinar of this webinar series. I hope that this webinar series will promote scientific temperament among the faculty members, and their knowledge will be extremely useful while carrying out their research projects. And I hope that their research will be useful to the society. I once again welcome you all for the today's webinar, and all of us, let us make this webinar series a grand success. Thank you.
Thank you very much, sir.
Thank you, madam.
Now may I invite the principal of Vignan Pharmacy College, Badlamudi, Dr. P. Srinivas Babu, sir, to give his brief introductory remarks. Dr. P. Srinivas Babu, sir.
Thank you, Purna Nagari. Welcome to the first webinar on overcome challenges in the formulation of lipid-based nanoparticles and their present applications by Dr. Srikanth Garu for coming and sparing his valuable time with us, and especially this innovative idea which was created by our beloved chairman, Dr. Lau Rattu Garu, and all the participants, especially faculty of all the 4 institutions, all will be benefited for this good extraordinary topic on the current usage. We're still having a lot of applications on the nanoparticles, so which will be enlightened by today's speaker, and they have more advantages because the lipids have high biocompatibility and biodegradability. So, and also it is having nowadays, if we can take, there are a lot of applications, but now as Srikanth will be enlighten us all the things, but which is essential applications like cancer therapeutics, and even which is having more applications for the current COVID-19 vaccines, and which is also very enlightened, and it may be helpful for all the formulation faculty who is involved in this formulation department like pharmaceutics, which they can overcome these challenges, some of these on the lipid-based nanoparticles. So for these innovative things, I congratulate the Vignan University, the head of the institution, Dr. Jitendra, and all the faculty members for making the first webinar of this session in the month of June. So I hope with all the blessings of our beloved management, we will do continuously for the benefit of our faculty as well as students. So I wish you this webinar will make a true reality, and which may help for all the pharma fraternity as well as the students and staff members. Thank you. Over to Purna.
Thank you, sir. Thank you so much. May I invite the HOD of Faculty of Pharmacy, BFSTRU Guntur, Dr. C.H. Jitendra, sir, to give his brief introductory remarks. Dr. C.H. Jitendra, sir.
Thank you. A warm welcome to all of you. It's a great pleasure to address all of you in this occasion, and especially in this way. We, for the first time, are collaborating with all the pharmacy institutions of our Vignan group, and it itself is giving me a very great enthusiasm and experience. we have initiated this webinar series in collaboration with all our institutions under the direction of our management, especially our chairman, Sri Rattaya Garu, and our vice chancellor, Sri M.Y.S. Prasad Garu. And this is intended to inculcate research interests in our faculty, as most of them are literally away from the colleges for almost 1 year, and this pandemic is also exerting a psychological impact, which is severely affecting the performance, initiation, their zeal or motto to achieve or accomplish. All such sort of things are getting degraded day by day by listening to lots and lots of unfortunate news. And hence we found that such programs are very essential at this point of time to boost the morale of our faculty and to make them more motivated towards the research. And we will be coming forward with more such webinars in the near future or in the coming days. And we want all of you to show the same participation and get benefited from this. And I should give the credit to all the heads of the institutions and the faculty, coordinators, my fellow colleagues, because as soon as we have planned such program, initially without any hesitation, all the principals have made this possible, and they have motivated their faculty also to be a part of it, their students also to be a part of it. And I expect the same cooperation in the future also. In the same way, we will be also extending our cooperation to make this series remarkable to Vignan pharmacy institutions. Thank you.
Thank you very much, sir. With these introductory remarks, now may I invite Dr. P. Raghuveer Verma to give the bio sketch of the speaker and to start the session. Over to Dr. P. Raghuveer Verma from BFSTRU Guntur.
So it gives me immense pleasure to read out the bio sketch of Dr. Srikanth Kakumanu. And sir has completed his MSc Biotechnology from Acharya Nagarjuna University in 2004. And later he moved to Division of Nanomedicine, Center for Health and Disease Research, University of Massachusetts, Lowell, on a drug research assistantship to pursue his PhD during the 2005 to 2010. And sir worked on formulation research, which included molecule stability, micronization, liposomes, nanoemulsions, polymer encapsulation, and crystallization for the delivery of active pharmaceutical ingredients. Not only delivery of active pharmaceutical ingredients, sir also worked on delivery of gene-based products. And sir has worked extensively on the encapsulation of botulinum toxin for transdermal applications. And in addition to his formulation research, sir also was actually involved in physical and chemical characterization of nanoemulsions, formulations, molecules, and analysis of biological efficacy at cellular and molecular biology level. And also, he worked on in vivo animal studies involving xenograft mouse models of different types of cancer, rat models for bioavailability, and hamster models of dyslipidemia, early atherosclerosis, and combined hyperlipidemia and hyperglycemia. And after his doctoral studies, Dr. Sir moved to Covaris at Massachusetts as a senior scientist and business development associate at Covaris. So his Sir's primary objective is around technology development of Covaris patented technology into particle engineering applications. And Sir was also involved in design, formulation, and characterization of formulations and testing in respective pharmacological models. And Sir also used his previous cell culture knowledge in their reagent and application development related to genomics and proteomics at Covaris INC. And with 8 years of experience at Covaris, in July 2018, Sir has moved to Precision Nanosystems as a regional sales manager. At Precision Nanosystems, Sir's primary role is to work closely with scientists who design, formulate, characterize formulations, delivery systems, and facilitating successful testing in respective pharmacological models. Sir has handled projects related to ranging from preclinical to clinical stage formulation projects, and moreover, Sir has led Precision Nanosystems microfluidic based platform technology to scientific field to the innovators, and he has demonstrated command of scientific knowledge to engage in collaborative sales process. And Sir has worked together with field application scientists to ensure customer satisfaction in pre and post sales situations. And Sir is currently working as a regional sales director at Precision Nanosystems, and also as an adjunct faculty researcher at University of Massachusetts Lowell. And here as a faculty researcher, Sir is involved in teaching labs and theory courses relevant to pharmaceutical sciences. The research at present is around glioblastoma treatment using mRNA therapy and lipid nanoparticles, nutraceutical delivery using nano and micro emulsions. And with over 16 years of experience with nanoparticles and their delivery, Sir has both academic and industrial experience, published extensively in 15 plus research papers in journals of international repute, and also had international patents. From his accomplishments, it is evident that he is an expert in applying nanoparticle technologies to biopharma, nutraceutical, and cosmeceutical fields. So with this introduction, I invite Dr. Kakumanu sir to take over the session. Please, sir.
Hello, everyone. Thanks for the intro, and thanks for the kind words. And I would like to thank the opportunity to present. Thank each and everyone who is involved in giving me the opportunity to present. Apart from all the information given, I am the alumni of Vignan Degree College. I studied from 1999 to 2002 at Palakaluru. So I just want to say that because I just want to make sure that you know that I studied at UMass Lowell. At Vignan. After Vignan is highlighted, so I studied at university, MSc Biotechnology, but my bachelor's was at Vignan. And let's move, because it's almost like we have only 1 hour. Let's move into the presentation. Let me share. Can you see it?
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir.
Okay, thank you.
Yes, sir.
Thanks. Hope you can see the full screen now, right?
Yes, sir.
Okay. Yep. Today we will be discussing mostly around lipid-based nanoparticles and their applications. We have around 1 hour, apart from presentation, if you want to reach me, you are welcome to reach me. I will give my contact information to Jitendra, or right after the presentation, I will give my contact information. First, let's discuss what are nanoparticles, and how are they different from, because I just want to give some intro, and my presentation would be my 16 years of experience, like how I transitioned and how I developed confidence on lipid nanoparticles or lipid-based nanoparticles, or the whole concept of nanoparticles using in drug delivery. Because a lot of students are attending, so I will just focus around my development and my knowledge improvement. That will automatically give how particles, lipid nanoparticles have transitioned and what are the present applications and stuff. Let's start with the presentation. And what are nanoparticles, right? Like, say, a lot of people might think that you can take a big particle and change into a nano small particle by just micronizing. A lot of you are from pharma. You can just break down them and stabilize them using a polymer and make a nanoparticle. You call them as nanosuspensions. To some extent, they are also complex particle because you're going to coat with some kind of polymer, right? But as such, like, it is having it's still simple. It's a nanosuspension. It's just a small molecule, right? So to explain the complexity of a nanoparticle, this slide really gives the reason why we call lipid-based nanoparticles as complex delivery systems. Because you can see that when you're talking about aspirin, it is just one molecule made up of 21 atoms. And, as it transitioned, the complexity of understanding of the whole drug delivery transition, the nanoparticles came in, and you can see that a nanoparticle is 100K molecules, and it is made up of a lot of atoms. So as it is a combination of a lot of molecules, it's very complex to understand them, right? And it is very complex to develop them. Even a small molecule development, it takes years to develop, right? Look out, like when you're dealing with thousands of molecules combined, it will be more complex to develop, a lot of analysis, a lot of physicochemical characterizations, and it takes a lot of time. That's the reason, apart from the structure, it is very complex to develop. So that's the reason they call them as complex drug delivery systems, right? And so nanomedicine, how these are applied in nanomedicines, right? How these nanoparticles are applied in nanomedicine. Nanomedicine is nothing but using nanoparticles encapsulating some kind of payload, like it can be a small molecule, a protein, a biologic, or a nucleic acid, and treating some kind of disease, right? That is called nanomedicine. And generally speaking, a lot of people who work in nanomedicine, they tend to say that anything below 200 nanometers or around 100 nanometers are nanoparticles. There is a reason for that. But technically, mathematically speaking, anything below 1000 nanometers is called a nanoparticle. But the smaller is better because you can sterile filter, you can make sure that there are no viral particles and stuff. That's the reason you will see a lot of nanoparticle research is below 200 nanometers, right? And why they encapsulate or make these complex drug delivery system using these APIs? Reason for that is they want to protect. They want to protect API, right? And they want to control the API release. The API can be a small molecule, protein, or a nucleic acid. And they want to alter the API distribution. They want to control how the API is distributed into the system. Make sure that there is no toxicity, there is more efficacy and stuff. And they want to deliver the API to particular tissue where the cancer is, where the disease is, right? And these all help actually, by making a nanoparticle or a lipid nanoparticle of these API, they actually get protected, they actually get controlled in terms of release into the biological system. Ultimately, it will improve the therapeutic efficiency. This right side picture here, you can clearly see different kinds of nanoparticles, and you will see people using different names in cubosomes, different kinds of names to their particles, because they name their lipid particles with different names, because depending on the structure of the lipids you use, they orient themselves into different kinds of structures, right? That's the reason they label them the way they are. Like, for example, lipid nanoparticle, which is our major focus here, is having a solid core, right? Everything is lipids, but how it assembles depends on what kind of lipids you use. And then there is liposome, like it has a water core. We'll explain a little bit more. So basically, it can be an oil and water emulsion, like here, right? So oil core, and it is stabilized by a surfactant. There are different structure of lipid based nanoparticles, different structures, okay? And based on your payload, you select your desired structure, or sometimes you might be a structural nanoparticle structural researcher, like then you will come, or you are producing your own lipids and stuff, then you will come up with your own structure, and you will label them as you like, or as it is deserved, okay? So my background, as mentioned by patrons and everyone here, my colleagues, they said that I come from a nanoparticle background of 16 years, but I didn't start that way. Before 2005, my training was almost everything in cell culture, microbial genetics, and plant transgenics at BARC, and IICB Kolkata. I have done microbial genetics there training, and then Pasteur Institute of India, I have done some vaccine based training, and then prolonged training around and work around almost a year or 1.5 years around stem cell banking, right? Hematopoietic stem cell banking. So my training and techniques are, before 2005, is all around cell, how to grow a cell, any kind of cell. Even today, I can grow any kind of cell, right? But when I came to my master's and PhD program at UMass Lowell, the nanoparticle or the nanomedicine field is very novice, and it is booming. There are only few products in the market, and it is like Doxil and Ambisome, right? But they are not very well understood. Everybody doesn't have information. Academic research started, and they started thinking about targeting and all those things. I'm fortunate to start during the time when it was very novice, but I went through a lot of confusion and stuff, right, during those days. I will try to explain why at the end of these slides, initial exposure to nanoparticle slides. These are a section of slides. At the end of the slide, I will explain why I got confused during these initial exposure days, okay? Initial exposure, right? When I joined my PhD, one of our colleague here said about high pressure homogenization. That's what, I used to think, okay, you make a nanoparticle by pre-homogenizing the ingredients, excipients, and putting them through a high pressure homogenizer, right? And getting a nanoparticle or nanoemulsion or liposome. That's what I used to think, right? Because published methodology at that time, a lot of publications are around high pressure homogenization or ultrasound, right? And even Doxil used ultrasound. Initial Doxil method used ultrasound, right? And means they used high shear methods or solvent methods to make the nanoparticles. So that's my exposure. So that's where I started. So these are the two instruments I used initially, M110H, 110S. This is from Microfluidics Corporation. And apart from Microfluidics Corporation, these kind of instruments are provided by BE International and many others, and they perform the same. So how it worked, high pressure homogenization, right? You generally take your, so the initial research was around oil in water emulsion and water in oil emulsions, right? And then we make creams out of it, mixing them in creams. And depending on where you're going to deliver, like topical and stuff, you either make creams or oral, you leave it like, liquid. And if it is IV or intramuscular application, there are different ways. But irrespective of the delivery, we used to make a nano emulsion first back then. How we make nano emulsion? We just mix the water plus oil and emulsifier, stir it, and we use high speed homogenizer or high shear homogenizer to make it as a homogeneous emulsion, right? Oil in water emulsion. And the oil can be any oil, like vegetable soybean oil, which is very popular in pharmaceutical sciences, or we used to use rice bran oil, castor oil, different kinds of oils. And of course, it also has a surfactant in it, which is not mentioned here, that has to be stabilizing the oil particles that are being generated, right? And then after applying a homogenizer, high speed homogenizer, we apply into this inlet reservoir of the instrument, right? And then it goes into the high pressure mode around 24,000 to 28,000 psi, right? And then it goes into the interaction chamber. Interaction chamber has something like a microfluidics model for that instrument from Microfluidics Corporation, but the channels are very big. These are applied towards a lot of shearing and pressures. Pressures create shear and all those things. These shear forces create small oil in water emulsion globules in the range of 100 to 200 nanometers. That's what we used to call as nano emulsion. So this is a high shear or high pressure based method, right? This is one method I used. And then I used something called phase inversion emulsion method. Like, we labeled it as self-assembling nano emulsion, where it's very simple. You put your drug, which is API, and it dissolved in oil, and then you add your surfactant, heat it to 50 to 60 degrees centigrade, make sure your concoction is clear, and make sure that your API is dissolved, and then you add water, certain amount of water, right? And then you actually drive it into a cloud point, right? And then at certain temperatures, it goes into from oil and water emulsion to water and oil emulsion. And then when you cool it, immediately it comes back to around 20 to 100 nanometer particles, oil and water emulsion particles, which are very stable, right? And of course, the surfactant used in this method should be a non-ionic surfactant. Only non-ionic surfactant can go towards the path of phase inversion. This is one of the technology we used, right? So this slide clearly explains how these oil in water emulsions by phase inversion forms, right? It's a monobasic or biphasic mixture, oil, surfactant, and water, right? And then you mix it and you heat it, and it goes into this cloud point where you will make water and oil emulsion, but that is very temporary. If you cool it, either slowly cooling or a fast cooling, it will go into an oil and water emulsion mode. So, but if you fast cool it, that will create a quick and very stable oil and water emulsion, right? So this slide explains how this is part of my thesis back then, how how a self-assembling nano emulsion, which is around 20 nanometer particle would look like, and how a high pressure homogenizer based nano emulsion would look like. I think this is curcumin, and this is also curcumin. Self-assembling is also a curcumin, and HPH is also curcumin to my knowledge. If you see that self-assembling nanoemulsion is clear, and these particles are around 20 nanometers, and these particles can be altered by changing your oil to surfactant ratio. As you increase the concentration of your surfactant, say for example, one part of one part of oil and one part of surfactant, basically in 50 ml of water, right, would create around 100 nanometer particles, right, depending on the surfactant and stuff. And when you have one part of oil and 5 parts of surfactant, which is non-ionic surfactant, you will make 20 nanometer particles, right? These are all lipid emulsions. In case of HPH, right, like say, you all know, most of you know that if you use high surfactant, it is toxic, right? In case of HPH, as you apply high pressures, there is a very good opportunity that you can make nano emulsions at low concentration of surfactants. So in this HPH, we use 1-to-1 ratio, and we formed an opaque emulsion, whitish emulsion. Here, the color is due to the curcumin, right? These are all nutraceuticals. And then we applied the high pressure homogenization and SANE-based technologies to different cell culture models, different small molecules. Most of this time, I was using doxorubicin, paclitaxel, and different small molecules, and some less means like not less high stable proteins like insulin and stuff. These are not subjected to degradation, right? I was using mostly those. And even if I use some nucleic acids, they are very small, like say siRNA, they are not sensitive to degradation, right? In this case, like, I just want to show that this picture shows a decarbazine nano emulsion, this white color, and an unprocessed emulsion, which is just a combination of excipients, which is oil, surfactant, and buffer, and the drug, turned into some kind of brown color when you put it in the room temperature after 6 months. And in case of nano emulsion of decarbazine, it didn't change. That says that the unprocessed emulsion, here I'm calling it as macro emulsion, there is some degradation. And in nano emulsion too, there is some degradation, but not to an extent of unprocessed emulsion, right? But you can see that the difference by eyesight, you can see the difference between a nano emulsion and unprocessed emulsion. At 4C, both the unprocessed and nano emulsion was stable, right? And the right picture was a part of our melanoma animal study and epidermoid carcinoma animal study, where we made a nano emulsion of decarbazine and put it at room, I think at 4C. This was taken outside to take pictures, and we have taken unprocessed emulsion, just a concoction, and then you process the unprocessed emulsion using a homogenizer, means like a high shear homogenizer. It's not a high pressure homogenizer, just mixing, that we labeled it as homogenized. You can clearly see that a nano emulsion of decarbazine was stable. At that time, 15 years ago, we were very excited, because now it is not, right? Because it is very well known now why it is stable and stuff. And nano emulsion delivery of tamoxifen. So we applied these nano emulsion platforms, and we have IP during that time. Based on IP, there used to be a startup in our lab itself. The company back then was called Encapsion, right? Apart from that, company using our technology used to focus around Botox delivery, which we used to develop on the formulation. But we used the same technology to encapsulate different small molecules, protein and stuff, and I will present few. Here, where we made a nano emulsion of tamoxifen and we tested on breast cancer cell lines. This is published. You can really get this article with this, this information here. We have shown that a nano tamoxifen is killing the, I think, HTB20 cells. These are HTB20 cells, not MCF7. We have noticed that it was a suspension is just a macro emulsion. A nanoparticle was actually influencing the cell proliferation, and it was killing the cells. And we have done extensive microarrays on these cells using Whitehead Institute, and we have shown that some of the molecular pathways are, I'm not an expert in microarrays. We used others like Whitehead Institute, and we have shown that a nanoemulsion of tamoxifen was activating some of the needed molecular pathways and deactivating some of the pathways that are not needed. Back then, it was a big thing because personalized medicine was very early, and we filed one of its first patent saying that nanoemulsions can do personalized treatment. Unfortunately, the research was not continued, and the IP department at UMass Lowell felt that that's not innovative back then, and they didn't extend the patent, and immediately a lot of people filed an IP around it. So this particular paper research showed really good effect at cellular level. But when we did the animal research, right, this is the confusion part. 15 years ago, 16 years ago. So we, as a researchers, we didn't really think about how this small molecule is working. All this chemistry and stuff is not that much. We are only focusing around how we are making the particles, but we are encapsulating it well or not, all those things, encapsulation efficiency and all those things. But there is a lot more. You need to understand the science, how these molecules work. I didn't even know that it is very cell specific. It is working on these cell lines, and it might not work on other cell lines. There are several physiological phenomenon that are needed for a nanoparticle to work. It just doesn't work by itself. There should be some support from the system, biological system, to take it and work. So this is a confusion, one of the confusion I had. This study worked really at cellular level, but when we did the animals, animal model, nude mice model, the tumors developed all over the body. And we couldn't really come up with a very quantitative assessment. So we have to shut down that study, right? And then we applied the nanoemulsion generated by high pressure homogenization towards a melanoma drug called it's a skin cancer drug called dacarbazine. And we published this work, and we have initially, right, we actually just took the cells and we dosed the cells with nanoemulsion, just like tamoxifen, and it didn't work. In cells, it didn't work. But at molecular level, we have shown some, by using microarrays, we are seeing some effect of nanoemulsion. So we went ahead as planned with our animal studies. That is the publication. It doesn't talk about cell culture. It talks about, animal studies. And we have shown that nano topical was influencing a little bit, means we create the tumor, we let the tumor to grow until certain millimeters, and then we will stop, we'll divide the animals into groups, and we'll make sure that all the tumors are in the same range, right? And we divide the groups of animals into treatment groups, where one group will be delivered with nano intramuscular. It's a localized delivery where you have the tumor, you tend to deliver it there intramuscularly. And then there is topical, where you just apply the nanoemulsion mixed with a cream onto the tumor. And then there is a suspension, which is not nano, direct injection, intramuscular injection. And in the similar way, you mix a suspension with cream and apply it. And we have shown that intramuscular injection was really effective. Actually, it completely removed the tumor, right? It was really encouraging, but it's still the confusion, why it didn't work in cells, right? And then we did the same formulation. We took the same formulation, and it is also published. This is the article. And we have done a xenograft mouse model of skin cancer. And we actually showed that it worked, particularly intramuscular nano dacarbazine was decreasing the tumor size really well, right? So these are the pictures from my study back then. This is it's not a nano, it's like a IM, intramuscular dacarbazine injection. You can see that it was actually creating wounds on the animal, This is a control where we didn't really treat it with anything. And this is the IM injected nanodecarbazine after 40 days plus 12 weeks. We had to sacrifice these animals because, they doesn't allow the animal to be there, because they don't want the ICOC and stuff. They don't want animal rights, like they don't want the animal to suffer. So after certain size of the tumor, we had to sacrifice the animal, right? But we have shown that, okay, nano emulsion technology works, and it works in breast cancer models at cell level and molecular biology level, and we have tested in animals, and it worked. But there are some gaps. It doesn't work for nanotamoxifen. It didn't work in animals, right? But in this case, dacarbazine, it worked in animals, right? So there is a lot of confusion, right? The confusion back then, I didn't understand, frankly speaking. I started in 2005, and I didn't understand, and it took me 2 to 3 years to understand. The reason for that is dacarbazine is a prodrug, and it didn't work because it has to be having some microsomal system to change the prodrug into active drug, and that was being supported by animals but not the cells. So we changed the cells, like say HepG liver cells, where the microsomal system is very active, and we have shown the effect that it was killing, nanoemulsion of dacarbazine was killing the cells more effectively. And we have also tested this concept taking with the help of Abbott back then. We have tested nanoemulsion of dacarbazine in microsomal system in vitro, and we have shown that a nanoemulsion was being metabolized more actively, almost 50% more actively compared to a suspension which is not nano of dacarbazine, right? Ultimately, I was confident, okay, I could figure out the reason why it is working in cells and not working in animals in case of tamoxifen too, because I think in case of tamoxifen, it's a different thing. I think the tumor model creation was not right. We didn't do it in the right way, right? So we selected a wrong cell line and all those things. We couldn't really create a localized tumor like the picture which you are seeing here, right? So that was the main reason. If we had created the localized tumor, that would have worked too. So the tumors developed all over the mice in case of tamoxifen nude mice model. Okay. So then we applied the same technology to the topical delivery of aspirin, where we have taken a lot of animals and created a inflammation model where we have our own internal technology back then to create inflammation in mice using croton oil. And by applying to the earlobe, it becomes thick due to inflammation. By treating with creams or aspirin suspension, nanoemulsion suspension, and we actually measure the thickness of the earlobe of the treatment after a certain time, like every 15 minutes or 30 minutes or 1 hour. And we have shown that aspirin nanoemulsion was actually, this was published from our lab, aspirin nanoemulsion was affecting the earlobe thickness more better than blank nanoemulsion, which you don't have aspirin. And even aspirin suspension worked, but not to extension of nanoemulsion of aspirin, right? And we didn't have any information on why it is working back then. We just used to make it and try it and look into the molecular pathways, and we know it is working, but we don't know why it is working back then, right? And then we moved the topical concept. I told you there was a startup in-house called Encapsion. So they used to deliver Botox. Botox is all these actors and stuff to take out the the old age symptoms of the skin. They use these Botox a lot, and this Botox actually is not good, but they use it in the industry a lot. This Botox is applied onto the skin intramuscularly to paralyze the muscle and to take out the folds in the muscle in the skin, such that they think that people will look young because there are no foldings, right? It was very painful intramuscular injection, and this company where we made these formulations was focusing around nano emulsion delivery by topical, just by cream delivery, which is less painful, right? And before doing this, because getting a Botox is very expensive, we don't have supply, and it is a toxin. It's a biologic, it's a botulinum toxin, right? So we didn't have supply of it back then. We have to collaborate with the researcher, and it took a lot of time. In the meantime, we tested with aspirin, we tested with insulin, and a lot of other molecules, which are proteins, and we tried to do a topical delivery, right? We got positive data in a lot of these things. So then ultimately we collaborated with someone, my professor contact at UMass Amherst, and he was able to synthesize a Botox, and the whole company went up in terms of research, and they tested in mouse muscles. Next slide. Nanoemulsion delivery of water-soluble substance, the protein Botox. You can see that this is a muscle picture. The black staining represents the release of acetylcholine esterase in mice at neuromuscular junctions. This actually induces paralysis, right? Which is the reason how Botox works. And you can see the intramuscular injection and transdermal injection is comparable compared to control, right? So it is transdermal delivery was showing some effect. Okay. So next slide represents the quantitative assessment, right? So nanoemulsion, intramuscular injection, nanoemulsion, topical application is comparable, right? So the company was focusing around topical because it is painless, right? And then the company spinned out of my lab and labeled itself as Anterios. They changed the name from Encapsion to Anterios. And around 2016, after 6 years of my graduation, the company was sold for $90 million to Allergan. This is the largest ever IP deal that was done by UMass Lowell. So this actually, until now, I was even in the phase of developing confidence, right? This has boosted my confidence to 100 plus, saying that there is something with nanoparticles. It will work. Even then, I had a lot of doubts because of targeting, targeting, even if you develop a targeting particle at bench scale in the lab, it is very tough to take it to the clinic, like, scale up and stuff. I had my doubts, but it boosted my confidence to a larger extent, right? So this, explains my confusion around why it is working in cells, in some cells, and it is not working in some cells, right? It's obviously physiological phenomenon, and it is formulation specific. And I didn't know back then. I had that confusion when I finished my PhD, why it was working in animals, why it was not working in cells. I still had some confusion, but I have some clarity on why it is working, right? But I had a lot of confusion, right? So when I went into the industry, I still kept in touch with academics. I teach at school and I do academic research even now, right? So during my industry interactions, that's when I came to know the potential of nanoparticles. First potential was not even a lipid nanoparticle. It was more just taking like a small molecule and making it a nano suspension by micronization. I know a cystic fibrosis drug went into clinic, and it is in the market now because of the concept of nanoparticles, because they could test this small molecule drug at a very low concentration, and they could test this small molecule at very less concentration of the drug available at the discovery or synthesis phase. The drug was available at very low amount in quantity. Actually, for doing a preclinical dosing study, to do the tox studies and stuff, they require certain amount. It was a lot below that, but using nano technology, they were able to do with meager amounts, and where they were able to move that molecule further into clinical studies and approvals, right? So that's when actually all these stories actually boosted my confidence. But I still have that how targeting will happen, how these cells, these nanoparticles would specifically target the cells, specific cells. I didn't have any expertise back then, or clarity on that aspect. Around 2016 and after, I actually started seeing, lipid nanoparticles, cationic ionizable. Actually, cationic or anionic surfactants or lipids, I actually came across in my PhD days only. But the thing is, those materials are toxic. you cannot use them, and the viability of cells is very low, right? So I still had that confusion. Even if they target cationic lipids or surfactants, they have their own toxicity issues, right? So I had that confusion. So after 2016, I started following papers from Peter Cullis, and started following very strongly around a lot of gene therapy companies that are being established around Boston, where I am. And I started looking into a technology called lipid nanoparticles, which can target to cells. And that actually has improved a lot due to pandemic. Actually, pandemic is bad, COVID situation, but pandemic situation boosted the whole LNP technology. The COVID vaccines of Pfizer, BioNTech, and Moderna are based off LNPs, lipid nanoparticles. The ease of scale up, the ease of manufacturing these particles, right, has actually made those vaccines available in a lot of countries like America and Europe and Canada very easily, right? So that's how I actually developed, confidence and knowledge towards nanoparticles, particularly lipid nanoparticles. Okay. So until now, if you see, a lot of my research was around small molecules and proteins, right? And this is when, from now, after 2010, I actually started getting exposed towards, nucleic acids and proteins that are, or peptides that are more sensitive to degradation. How can you encapsulate them without degrading? My previous technologies, whatever I explained, cannot encapsulate these molecules very easily. Even if they encapsulate, they will degrade certain percentage of these molecules. I want 98% encapsulation, or the industry wants 98% encapsulation, because it's very tough to synthesize these molecules, right? And back then it was very expensive to make these therapeutic molecules. Even now they are very expensive, right? That has moved me towards, I still worked after 2010 with high shear methods to at low power to control using focused ultrasound and stuff. Still there are shear forces and the molecules are degrading above certain, for example, DNA around above 2 or 4 kb was degrading. How much low energy you apply, it was degrading, right? So that was, I did those experiments by myself. So I kind of recognized that, okay, heat, shear forces is no go, right? And around 2010, I actually interacted with his name is Chris Ho at Genzyme. Back then, he's the director. I was in lunch with him during a visit to his company, and he told me during the lunch, Sri, looks like you work a lot on small molecules. Small molecule is no more an innovation. It is biologics. Even proteins are going to go away. It is genetic medicine. Means people will be trying to deliver genetic material like mRNA, siRNA, delivering cells itself, right? Cell therapy. He told me that. I was right out of school back then, and I did get his piece of advice into my mind and started working towards it, right? And he's absolutely right. If you see now, in last 1 year or so, $20 billion are invested into lipid nanoparticles or gene therapy market, right? My insight of lipid-based nanoparticles 2010 and present will be presented here, right? Why, why you need lipid-based nanoparticles for, it is now, as of now, it is a lot used in RNA delivery, like our plasmid delivery, which is very popular in genetic medicine and cell therapy. Why it is used? So even there are other options to deliver this genetic material into the cells. One is electroporation, right? Which is using electric potential. They open up the cells and they introduce the genetic material. These methods come under mechanical methods, and there is sonoporation where they use ultrasound to open up the cells and and put the molecule into the cell. There is something called squeeze technologies, where they squeeze the cell to open up, and they introduce the not small, you can introduce a small molecule to, like any molecule, into the system, right? And obviously there is a gene gun method, and all these methods are there, but all these mechanical methods are okay at small scale, at R&D scale. They cannot be scaled up. You need to work with the technology that can be taken into the clinic, so that they can do the clinical studies very well, and they can release the drug under regulations into the market, right? Mechanical delivery method is not scalable, right? And the payload is not protected, it gets degraded, right? And then there are these viral delivery systems like AAV and lentivirus. These are good, they really work below 3.5 kb, for example, with nucleic acid. But nucleic acids, mRNAs, they are trying to deal with 4 kb and above. Some people are dealing with 10 kb kilobase pairs. Some people are dealing with 20 kilobase pairs, right? So it's very tough to. So they have to break the molecule of delivery, and they need to deliver the molecule in bits and pieces using viral vectors. So that is not efficacious. And then even viral vectors, potential immunogenic effect. Obviously, it's a viral particle, right? It has immunogenic effects, and scaling up the technology is very tough. A lot of stringent regulations should be taken care, and it's not easy to take a viral delivery system into a human biological system, right? That's when these were developing parallelly. Chemical methods like liposuction uses cationic lipids, right? And there is a calcium chloride methods. All these methods also cannot be scaled up easily. Even if they scale up, liposuction or cationic polymers, they actually tend to be viability issues. It kills the cells, right? Like mechanical and viral methods. So if there is no viability, right, you are not going to deliver it effectively. So you need to have, when you deliver, the cells should be alive, and then the genetic material has to do its course of action in the cells, right, to change that cell into healthy or just remove the unhealthy cells, right? So that's when lipid nanoparticles came into existence, where in case of lipid nanoparticles, instead of using cationic lipids, they use cationic ionizable lipid. They can control the ionizability of these lipid. At certain pH it will ionize, at certain pH it will not ionize, so such that it can be controlled in terms of toxicity, right? These are very well now used. Almost everybody is working with these lipid nanoparticles, right? So, and these are very well designed because of their low toxicity, low immunogenicity, and manufacturing capabilities. And the matter of fact, it is very robust. It can deliver miRNA, siRNA, mRNA, CRISPR guide RNA, everything. So it has impact in all these genomic technologies. So it is going to be going big. So it's good for you to get exposed and know more about lipid nanoparticles, right? So just a little bit explanation of different structures, which I'm going to focus. The right dense core structure is a lipid nanoparticle, right? And this lamellar structure is a liposome, right? You can see a water core, right? And these are the excipients, positively charged ionizable lipid, PEG lipid, DSPC, and a neutral ionizable lipid, and then cholesterol and siRNA. Sometimes you might not use positively charged ionizable lipid, and if you don't use that ionizable lipid, you will make a liposome, this structure, right? You can see next few slides how the liposome is formed, or how a lipid nanoparticle is formed. So this is anatomy of liposome, right? And you can clearly see that it involves a PEG lipid, and it prevents ostwald ripening. Ostwald ripening is your particle imbibing water or buffer in the solution, from the solution, and then it increases and actually it destabilizes. That's called ostwald ripening. PEG lipid helps in stopping the ostwald ripening. And then there is helper lipid. It forms bulk membrane, increases fluidity of the membrane. And then the cholesterol, it increases the rigidity of the membrane. And you can, using liposomal structure, because there is a water core and there is a hydrophobic or amphiphilic cell, like like a bilayer, which forms, which is similar to a cell, right? A liposome is a cell without life, right? Literally, you can compare the center core as a cytoplasm or something like that, and then this is a cell membrane, right? In the membrane part, you can actually encapsulate a hydrophobic drug or an amphiphilic drug, or in center core, you generally encapsulate a hydrophilic drug or a the hydrophilic drug can be a protein or a nucleic acid, or you can, even in small molecule which are hydrophilic can be encapsulated. So these nanoparticles can be made by by the methods I showed you, high pressure homogenization and so ultrasound methods and stuff. In next few slides, I will introduce a less stringent method where there is no shear forces and stuff. So this is a liposomal structure, and next is this is a LNP structure. What is the difference? You will see it has almost same ingredients. The biggest difference is this ionizable cationic lipid. This ensures high cationic, and nucleic acid is anionic, right? The cationic lipid and nucleic acid anionic nature, right? Phosphate and NH4 groups, they combine, and that will actually increase high encapsulation efficiency. I was never doing this before, right? So this cationic ionizable lipid, I was doing with cationic lipid, but they are toxic. Ionizability is the biggest aspect, right? And then the rest is the same. You will see in cationic, in LNP structure, you will see this ionizable cationic lipid. That is a big difference. You will see cholesterol, helper lipid in a liposome, PEG lipid in a liposome, right? And you will see a payload too, right? The biggest difference is in a LNP structure, you will see a cationic ionizable lipid that will actually create this solid core instead of the water core, right? Hopefully you understood what is the difference between liposome and LNP structure. Okay. So LNPs are used for the time is running. I think it's already 7:50. I have a lot of slides. So LNPs for effective nucleic acid delivery. Why LNPs are used? It's just an overview, right? It can be, as I told you, it can encapsulate very well with almost 95, 98 encapsulation efficiency. Obviously, it's efficient in delivery. Why it is efficient in delivery? So it can, lipid nanoparticle can interact with LDL receptor very well, right? In cell culture, if you see a lot of papers, they tend to add ApoE, which will help lipid nanoparticle in interacting with the cells, right? So these ApoEs will interact with cholesterol, and then they will go and interact with LDL receptors really well. Yeah. This this is not like completely proven, but a lot of people believe in this. It's like EPR effect, enhanced permeability retention effect, how a nanoparticle works. Back then, when I graduated, when I presented that, my thesis advisors said that this is not acceptable. But now everybody believes in enhanced permeability retention effect. Small molecule, small particles like nanoparticles retaining in tumors more efficiently than bigger particles. So that is enhanced permeability retention effect. That happens because of the physiology of the tumor, right? So that was not accepted back then 10 years ago. Now it is accepted. So in this case, lipid nanoparticle, how it is endocytosed. So obviously it is going to use LDL receptor and gets endocytosed, and change in pH in the cytoplasm will help in endosomal escape means engulfing of this lipid nanoparticle into the endosomal system will be avoided. But this lipid nanoparticle will interact with the phospholipid layer of endosome and will break out and release the nucleic acid or your payload into the cytoplasm to take part in, protein synthesis pathway. Okay. Sometimes you might see some people delivering plasmid DNA with or DNA molecule with this methodology. The only thing lipid nanoparticle will do is releasing the payload into the cytoplasm. From cytoplasm into the nucleus, because for a plasmid DNA to work or a DNA to work, it has to go into the cytoplasm, into the nucleus. That is not done by a lipid nanoparticle. Lipid nanoparticle efficiently, and it is very well proven that it is delivering it into the cytoplasm very efficiently, right? So evolution of formulation methods, like I actually talked a lot about high energy methods, and what are the problems, high shear forces and harsh conditions, and difficult to scale. Very large scales are very difficult, right? And then people tend to transition into inline mixing. Even inline mixing used to create a lot of macrofluidic mixing, right? And it used to degrade the molecules, and it's not efficient in scale up, right? It's a gentler, you have to go for a gentler process conditions, and then it will not really create a very good mixing method. That's when microfluidics is there in the system from long time. It is applied in life sciences from long time, but that's when actually people started applying microfluidic approaches. Precision nanosystems does that to make these lipid nanoparticles or liposomes or any nanoparticles, right? It's a very simple method. That's the reason it is very easy to scale up, right? It is very easy to scale up, reproducible, and this actually helped the microfluidics technology to go into clinic very easily, right? And this explains, the slide explains a little bit about how microfluidics technology mixes the different materials. These colors represent different materials, and in a microfluidic environment, it's a laminar flow mixing. You will not see any turbulent mixing. They have to fold to themselves. To fold to themselves, you need to create environment in these microfluidic channels so that these layers can fold on one onto others and create good mixing method. Coming to macrofluidics, which is turbulent flow, it creates, on the right here, it creates a lot of shear forces. This can degrade your sensitive molecules like mRNA and all those things, right? This picture explains about how these nanoparticles, lipid nanoparticles, liposomes, or even PLGA particles are made, right? You will have a solvent where your lipids are dissolved at certain concentration, and you will have an aqueous. If you are having an mRNA or any hydrophilic drug, it will be dissolved in your aqueous buffer. And these are combined at certain ratios, right? And at certain flow rates. And you can see that when you get out of the outlet, you will get a nanoparticles. This is a pure precipitation. There is no shear forces, nothing. It's just general mixing, right? And Precision Nanosystems has two technologies based on microfluidics, which is classic SHM, a lot of publications, around 250 publications on that. And then they have this next gen technology, which can be scaled up very easily, right? So people have transitioned so that, Pfizer is making, millions of doses. Means people have transitioned these technologies to make lipid nanoparticles or at a really larger volume, so that it is even catering the pandemic situation, right? And it is a little bit more explained here. Lipids are dissolved in a ethanol. The popular method is ethanol injection method. Don't use any other solvent. Try to use ethanol and non-harsh solvents. I tend to move, when I started my research, I started using chloroform and all those things. Don't use those. Try looking into something called ethanol injection method. Ethanol is very well accepted in regulatory filing, right? And if you need to remove it because you need to make your particle stable, but ethanol is not a harsh solvent. And it is also the matter of fact that you can use ethanol to dissolve the lipids and make these liposomes, lipid nanoparticles, is actually helping even the scale-up process more easily, right? So look into a method called ethanol injection method to make lipid nanoparticles or liposomes. You will have a lot of information. And this is a very simple pictorial representation on how it is made, right? Again, lipids or hydrophobic stuff are dissolved in ethanol, hydrophilic stuff are dissolved in buffer, which is very important, and they are mixed with a non-turbulent flow, and then the particles come out by simple precipitation, right? The only thing these mixers are doing is good environment for mixing. And it has to be controlled and reproducible, and there are several process parameters. Obviously, by the picture you will know that you're pumping these solutions, the solvent phase and buffer phase. There is flow rate, right? There are ratios. You need to really work around that. What are the ratios? And initially, when the particles start forming, there might be some waste. You need to know how much you need to take out as waste, right? So that you can control your physico-chemical characteristics of your particles. Basically, if you don't remove that waste initially, which is not really good particles, your polydispersity index will be very broad in many cases. So that is the reason. In some cases, that might not be, so they will not have waste in their process. So you need to really figure out all those things, right? And if you work on all these process parameters and chemistry parameters, you have your good lipid nanoparticles, right? After getting a good lipid nanoparticle out, what you still have solvent, which is ethanol, right? You need to remove it, right? At a bench scale, what you are looking at is normal flow, like normal flow filtration, these filters where you can use in centrifuge and all those things. These use normal flow filtration, and they are fine at small volumes. But when you go to larger volumes, scale up, non-human primates or clinical studies, you are going to make larger volumes. You cannot use normal flow filtration. The reason is, it is very well explained in pharmaceutics, that it actually blocks the membrane, and it cannot be scaled up very easily. But in case of tangential flow filtration, the flow will not allow these particles to accumulate on the filter, and then the filtration is more seamless comparatively. Okay, it has its own complexity. If you just know tangential flow filtration, you have a job in the market. Making a nanoparticle is simplified by companies like Precision Nanosystems, particularly Precision Nanosystems. And tangential flow filtration is the most complex workflow in the whole workflow of making a nanoparticle and putting into the clinic. Is the biggest complexity is around tangential flow filtration. It's very complex, and if you have a knowledge in that, and if you work towards that, you will have a job when you are right out of into the industry from your school, right? This slide explains about how a normal flow filtration works and a tangential flow filtration. Due to the timing, I might not cover, right? So I explained in the previous slide. And factors to consider to what kind of method, filtration method you need to use. Obviously, physico-chemical characteristics, like you need to keep the target product quality attributes like particle size, polydispersity index, and encapsulation efficiency. Apart from that, what is the volume, right? And you are going to work, small volume, you can go with just normal centrifugal filters. Larger volumes, you need to go for tangential flow filtration. And then what is the payload you're going to encapsulate? Is it a nucleic acid? Is it a small molecule? All those things. What is the solvent you used? All those things should be really understood before you select a filtration method. Okay. And then a lot of papers will come across this. You are making a good particles, right? To make sure that you are making good particle, right, you need to generate data, particle sizing, right? Dynamic light scattering is very popular method in that. Malvern Instrument is one of the company that provides. Apart from that, there are a lot of other companies that provide these instruments to measure the particle size. They measure the particle size by Brownian motion. It has its own flaws, that it will assume any particle as circular. So it is fine for a lipid particle, because they tend to, most of the lipid particles that are used are circular, or near to round in shape comparatively, right? So basically, it gives out the Z average and the polydispersity index, which is the size distribution, dynamic light scattering. And then you will come across encapsulation assays, and these are payload specific. In case of mRNA, you will come across ribogreen assay. There is a lot of information online. You can go and pick it up. And there is, if you are doing with the plasmid RNA, plasmid DNA, they call it a picogreen assay. And small molecules, as you know, they use HPLC. So all these tell encapsulation efficiency is nothing but it will tell how much of your drug is there in your 1 ml of lipid nanoparticle or nanoparticle formulation, right? And this slide says about liposome drugs. As of 2017, there are 15 drugs. I don't know about all of these drugs, but I have worked with cancer drug called Doxil and its generic format before 2016. And I have also worked with Ambisome, which is used in fungal infections. Ambisome is amoxicillin liposomes, right? So there are other places where these liposomes are used in vaccine, analgesia, and macular degeneration. Unfortunately, I didn't work with liposomes in that field, but I have worked with Doxil and I have worked with Ambisome. Both of these patents are expired, and Doxil expired a while ago. There are a lot of molecules that came, a lot of research that is happening to come up with a generic. And in the similar way, Ambisome, a lot of people are trying, and some people are successful, right? So that's an opportunity when you go out from your school to industry. So coming to the lipid nanoparticles, I'm going to talk a lot about lipid nanoparticles, but they are not, there's only one drug, one patro from, it is also called as Partisaran, and, it is from Alnylam Pharmaceuticals, and it is the only approved drug. And then you see Moderna and Pfizer COVID vaccines, but this one patro, this is the centerpiece. It can be called as mother of all inventions, right? This has helped further LNP research. Onpatro came up several years ago, but even then, regulatory pathway is very stringent. The only reason you see Moderna and Pfizer drug, COVID vaccine based out of lipid nanoparticles is because of pandemic situation, urgent need, right? So they have released it without doing all the general regulatory pathways, right? So they have done a lot of toxicity studies. They have tested in humans, right? But they take more time comparatively. They are very quick because of the LNPs and the matter of fact that LNP technology is available. So I was going to discuss about more in depth about LNP and liposome structure, right? But it's already 8:04. I'll try to skip and tell you about few things in this LNP liposomes and the considerations you have to take when you are going to make a lipid nanoparticle, right? So this I already explained, what is the structural properties, what is there in a lipid nanoparticle, ionizable lipid, what it does, right? Neutral at pH 7, cationic at pH 4.4. So when you are encapsulating, right, it needs to be cationic, right? So your buffer should be low pH, right? And when you are not encapsulating, your particles are formed, you have to reconstitute your particles into a pH 7 buffer so that you avoid any kind of disassembly or instability, right? Stability issues, right? And it explains again what does the cholesterol does, helper lipid does, right? And peg lipid does. They have very important aspects like stability, structural characteristics, and everything are influenced by all these ingredients. Cationic ionizable lipid helps in delivery into the cells. The rest of the things are structural, and they are also involved in attaching the lipid nanoparticle onto the cell membrane and stuff, right? It also gives the stability and stuff, right? So again, formulation of a nucleic acid based lipid nanoparticle, you need a payload, which will be an mRNA, siRNA, plasmid DNA, right? And then you will have excipients, which is your lipids. And then you will have a formulation medium, which should be a buffer. That is very important. The pH is very important, right? And then there is a formulation method. You just inject into a microfluidic platform, and at certain ratios, at certain flow rates, optimized flow rates, and you get your nanoparticles around 80 or 120 nanometers, right? That the particle size depends on your formulation, right? So this explains a little bit more about the payloads, siRNA, what would be the size, 21 to 23 base pairs. mRNA is 200 to several thousand base pairs. Plasmid is 1 to 200,000 base pairs. So basically, you will think that, okay, as I increase the particle size, as I increase the molecular weight of these, because you can see that miRNA and sgRNAs are small, smaller than the plasmid, and siRNA is very small. You might think that as you go increase in the size of these molecules, you might need to increase the concentration of the lipid, to some extent, little, but not too much, like a small molecule encapsulation. For nucleic acid, there is not much change in the lipid concentration. What you need to change is you need to work with your buffer pH, and you need to work with buffer ion concentration and stuff. So apart from that, you need to slightly increase the lipid cationic ionizable lipid concentration. That's it, right? So cationic, I explained, they are toxic, and it binds to nucleic acid. It will be toxic, but ionizable, right? Low tox, and the toxicity can be managed, right? And you can make a homogeneous structure like that, and using a toxic cationic lipid, you will be making a more like a lamellar structure, which is more similar to liposome. Okay. One important thing when you are encapsulating a nucleic acid that you need to take care is N/P ratio. N is on your lipid, NH4 group, positive group, and phosphate group is obviously on nucleic acid. This actually ratio is very important to encapsulate a nucleic acid, right? So you can clearly see that in this pictorial representation. In the next slide, you can clearly see if you have adequate NH groups for phosphate groups, your nucleic acid is protected. If you don't have NH enough NH4 groups for your nucleic acid, means it's not ionized properly, your lipid during encapsulation, so it will not encapsulate, and obviously it will not protect your nucleic acid in the biological system or in buffer. Okay. So I actually tried to explain what is N/P ratio, and but we can talk about due to time constraint. I'm not going to cover a lot of it, but N/P ratio is very well explained in my previous slide. It is NH4 positive group from the lipid, cationic ionizable lipid, and phosphate group from the nucleic acid. There should be enough NH4 groups available for the phosphate group of the nucleic acid to get it encapsulated. The better the ratio, the better they are. Generally, around 3 to 4 or 3 to 6 N/P ratio is very good, depending on your nucleic acid size. You might change the N/P ratio. So basically, this slide explains about how to calculate the N/P ratio, right? So, but I can share this information if you are interested. You can send me an email on how to do a N/P ratio. So for the sake of presentation, just take in terms of N/P ratio is very important for efficient encapsulation efficiency. And N is part of your lipid structure, cationic ionizable lipid. Phosphate is part of your nucleic acid structure. And availability of these groups are very important for proper encapsulation. and the ratio between N and P groups is N by P ratio. Okay. So this slide explains about what happens if you have a peg and if you don't have a peg. No peg, the particles will collide to each other, and they will ripen and become big particles. But if you have a peg, it will influence steric hindrance, and it will actually help the particles to be separate, and it doesn't allow the particles to ripen, ostwall ripening and all those things. So it actually stabilizes the particles. This slide explains that. That is very important. Having a peg in your peg lipid in your formulation is very important, right? And this slide explains about percentage of peg lipid. Higher the percentage of peg lipid, the smaller the particles you will get, right? If at all when you are formulating, your particles are bigger in size, and if you want to decrease the particle size, increasing the concentration of peg is one of the option. Okay, this is an electron microscopy picture. And apart from that, depending on one of our colleagues during the introduction said that there are different kinds of particles, right? How these different kinds of particles are formed. These different kinds of particles are formed because of the helper lipid generally, which helps in forming these particles and bulk structure. Depending on the structure of these lipids, it can form a hexagonal phase or it can form a lamellar phase. So these helper lipids drive the structural formation of lipid-based particles. Okay, and these lipids can be, ionized. Sometimes they cannot be, means ionic. Sometimes they can be non-ionic, right? So importance of pH, generally when you are encapsulating, it has to be at, your buffer should be at pH 4, so that your lipids can give that NH4, the positive charge. And when you make the nanoparticle and when you want to stabilize it, it is better you are at physiological pH 7, so that there is no more process happening, right? So this explains that, right? And this also explains the importance of pH and buffer strength, a little bit more in depth, right? If your lipid is ionized properly, it will have this association. All ionizable lipids are charged. You will have efficient encapsulation. If your lipids are not charged, no association at all. If your lipids are little bit charged, little bit of association. So all these is being influenced by the pH of the medium you are going to use, right? The buffer pH. Okay. And this slides talk about importance of sodium and potassium and NH4 ions, right? And and the matter of fact that why you need to avoid Ca2 2+ divalent ions, particularly cations, because it interferes with the nucleic acid encapsulation. So you will not get efficient encapsulation. So when you're making buffer, which is your liquid phase, buffer phase, where you will dissolve your nucleic acids, you need to make sure that there are no divalent ions like magnesium and calcium ions in there. And monovalent ions are good. So you can have sodium, potassium, NH4+, right? And so this slide explains about effect of mRNA length on mRNA LNPs. That's what I tried to explain in one of the slide. It doesn't matter the size of your mRNA or your payload. It will form the same particle size because there is a lot of NH4 groups still available on the nucleic acid on the liposome lipid to encapsulate further bigger size nucleic acid. So you can see that GFP mRNA 996 nucleotide length and luciferase mRNA almost 2000 nucleotide length and Cas9 mRNA 4500 nucleotide length. You can see that encapsulation efficiency is the same, almost 90%, and you can see that their size is almost same, not a big difference here. And even the PDI is not that much the difference. They are all around 0.1, in between 0.1 to 0.2. So this says that increasing the size of your payload has minimal effect on biophysical properties and encapsulation efficiency on mRNA, on LNPs. So this says that it's a very robust technology, right? And this slide talks about lipid-based particle applications. So as I first, lipid nanoparticles can be used in oncology. So oncology is part of gene and cell therapy. There are companies who are working with cardiovascular disease. They are working, some companies are working with acute macular degeneration, eye-related disorders, rare diseases, right? Genetic diseases. Apart from that, mRNA vaccines, right? Protein replacement therapies are all based on LNPs, right? And those are the applications. And here are the materials that are used to make a lipid nanoparticle, right? And in case of liposomes, these are the application: vaccine adjuvants, antimicrobials, cancer chemotherapy, and diabetics combination therapy. Mostly here with liposomes, I would say small molecules, proteins are the targets for encapsulation in terms of payloads, okay? And these are the carrier materials: phospholipids, cholesterol, PEG lipids. Clearly says the only difference between liposomes and LNPs is ionizable lipid, right? And then polymer nanoparticles, I have done considerable amount of work here. And this is a very popular field now because these polymers can be manipulated as you like. And there is a lot of research around, because there is a freedom of access with lipid nanoparticles, the cationic ionizable lipid, because of the IP, intellectual property, and all those things. So a lot of people are looking into polymer nanoparticles. I have done polymer nanoparticles, little bit of protein work, and I have done a lot of small molecule work, encapsulating small molecules and stuff. So emulsions, they're used in cancer therapy. And one of the popular emulsion is oil and water emulsion is restasis emulsion, a cyclosporin emulsion that is used for restasis, right? Dry eye disease. And there are organic and inorganic nanoparticles. They are used in imaging and theranostics, okay? These are the applications and different particle types. And I would really suggest you to really focus around nucleic acid based lipid nanoparticles. I think that's where the field is going, okay? So this is what the field is going. It's all about genetic medicine. I don't think you will see, you will still see small molecule research, but not a lot of investments that will go. So investments will create jobs. So if you want to have a job in, in future, I think genetic medicines and disease research around genetic medicine is really good. The genetic medicine works by silencing genes, RNAi, expressing genes, right? And editing genes, CRISPR technologies. So, being, well informed on all these technologies is good for you. Try to write some review papers, try to read papers. And, if there is funding, I would suggest you to start a program around genetic medicines at your school. So conclusions are, right, lipid nanoparticles solve key challenges, and it is clinically proven in the form of already approved drug on Patro, and COVID vaccines supports that, like, both Moderna, as I told you, Moderna and Pfizer uses LNP technology. These are very low toxicity, and they can be, they protect the payload to a larger extent, right? Very well, and it is delivered efficiently into the cell, and it can be controlled. The RNA or the payload release can be controlled really well using a lipid nanoparticle, right? Still, there are some challenges. As I told you, access to the ionizable cationic lipids is, if you have a lipid program, if you work around your own lipids, I think that's a very innovative way. A lot of people are working around their own lipids. For example, Moderna, they work their own lipids with their own lipids. Pfizer, BioNTech, they didn't have the lipids when they were making the vaccine, so they have to depend on a lipid manufacturing company called Acuitas. So the center piece is ionizable cationic lipid. If you are interested in lipid research, that is something I would suggest you to work around. You know, and and ultimately another conclusion I would like to do is, if you are going to work with LNPs and RNAs, genetic medicine, you need to really look into the lipids, the materials used, helper lipid, cationic ionizable lipid, whether you can make your own or in your synthesis lab, or procure, but they are very expensive, right? And how to optimize. It's very simple formulation, how the flow, like, you mix your lipids and buffer lipids in solvent and buffer at certain ratios and at certain flow rates, you get your particles. So you need to really think about the process parameters and how to optimize, right? Where to start is with, obviously, literature. There is a lot of literature. If you don't find any literature, please do contact me. I will be really happy to provide articles, and if you have any doubt, in future, I will be really happy to interact, right? So literature is a way to start. Please start with literature, and then if you have already knowledge, start working on the materials, right? And if you already know about the materials, start working on a disease target, right? And if you have already a disease target, right, then you are ready to go. You have a program, right? Grants and stuff, right? So, and if you need help in writing grants and stuff, I will be really happy to work with you. Thank you for listening, and thanks for the opportunity to present. Any questions?
Hello, sir. This is Dr. Raghuveer from Department of Pharmaceutical Sciences, Vignan University. So actually, I have a small doubt regarding these payloads. So whenever you are talking about the payloads, you are talking about mostly about these genetic material like mRNA or DNA, some stuff. So actually, I am a chemist, so I wanted to encapsulate certain small molecules. So what would be the payload, like in terms of mg, milligrams or micrograms, that can be encapsulated into a particular liposome or lipid nanoparticle?
So that's what, like I told you, right? Like, and I was in that field, like, say, 20%, 10% encapsulation, because in case of small molecules, there is no directed, right? There is no this charge effect like what we talked. So with these, there is less encapsulation. If you want a lot of encapsulation, obviously you need to increase your lipid concentration, right? That's when it's all dependent on your concentration, and there is a limit on how much high you can go on lipid concentration, because viscosities will raise and it will not process, and all those things, blocking your channels or blocking your process equipment will occur. So technically, I think there is a limit on how much you can encapsulate with small molecule. You cannot get 98% encapsulation. So
yeah. So maybe that might be because I was interacting with some of my friends in industry who are working on these liposomal systems, and they were always telling that it's very difficult to work on like antibiotic drugs for liposomal drug delivery. So thank you, sir. Thanks for clearing my doubt.
So one thing, just to go on offline, we can talk about, I worked with Doxil. So initially when they made liposomes, right, like in Doxil, they don't really make simultaneous loading. They make the liposomes and then they use those blank liposomes in a remote loading method. They load, the doxorubicin into the liposome, into the solid.
Something like the permeate doxorubicin to the liposome.
So they call it salting out, salting in method, remote loading. I have done, I didn't get like 90% encapsulation, but I have got like 75, 80% encapsulation. I'll be really happy to work with you if you are interested in that. So, I have the written protocols and stuff, what are the materials requirement and all those things. So I will be really happy to work with.
Sure, sir. Sure, sir. Thank you. Thank you very much. So, any questions? from the participants?
Sir, good evening, sir.
Yes.
Myself, Prabhakar Reddy.
Hi.
Sir, as compared to a traditional dosage form and a lipid-based nanoparticle, particularly the chemotherapeutic agent, sir.
Mhm.
So, how many folds the price is going to increase, sir? Normal idea.
I mean, like obviously, we got all LNP vaccines for free, right, because of the pandemic situation. But you are asking, like, say, for example, for a chemotherapy drug, say, if you compare with Doxil, which is doxorubicin liposomes, and if for breast cancer or oncology drug, if you have an mRNA LNP for oncology, what is the difference in the pricing? That's what you're asking, right? The pricing, they are controlled by the governments. They cannot put too much price on there. But obviously, they have invested billions of dollars. They will try to extract money from people who can pay, but the governments are there to subsidize and stuff. So I don't think I can comment a lot on that. But the thing is, I'm sure that it will be more expensive because the people, the innovator, like people like Moderna, they have control on mRNA, they have control on the payloads, everything is theirs. They synthesize and they make, even the manufacturing technology. But there are companies who depends on other lipids, so they have to license, they pay royalty. So if they pay $1, they try to get at least $2, right? So they have to, unless and then these technologies become free access out of warrant, IP and stuff, it is very tough to provide these at lower price. But they will become. It will be. Simple example is the generics, right? Dr. Reddy Labs makes a lot of generics, and they actually provide affordable medicine in the form of medicine, small molecules, to a lot of people in the world, right? In the similar way, definitely these will become cheaper, but they have to be accessible first, right?
Thank you, sir. Thank you very much for giving elaborate information on nanomedicine and various comparative studies. And thank you very much, sir.
Still, if anyone is having questions, please ask.
Sir, good evening, sir. Myself, Sri Rekha.
Hi, good evening.
I am audible, sir.
Yes, I am.
Sir, I am just registered PhD, and I want to do the project on solid lipid nanoparticles loading with the hydrophilic drug, sir. So one of the challenge in case of solid lipid nanoparticles is the entrapment efficiency of low entrapment efficiency of hydrophilic drugs. So how to improve the entrapment efficiency in case of hydrophilic drugs? Is there any method or selection of lipids?
So the thing is, sorry if I missed. Are you working with liposomes, or are you working with water in oil emulsions? What kind of payload, particle are you working with?
Solid lipid nanoparticles, sir.
Okay, you are working with
by
solid
loading with hydrophilic drugs. I want.
But solid core, solid lipid nanoparticles will have solid core. It will not give good encapsulation. So if you want to encapsulate a hydrophilic molecule, it has to be a liposome.
Okay.
So there are several other methods that can be done. Try moving towards having a water core. So it needs to have a loading capacity. So it needs to have that water core to load yeah. So to my knowledge, so I will ask you, suggest you to look into other lipid-based particles than solid core.
Okay, thank you, sir. Thank you.
Sir, I would like to ask one more doubt. So, as you are talking about this liposome, I mean a lipid core inside which a solution is present, a liquid is present. So, is it possible to measure the pH of the core liquid? Is it possible to measure?
Yeah, I think so. The core of the lipid will obviously the pH of the buffer you're going to use.
Okay.
So yeah, there is no need to measure, but there should be a way to measure, but I haven't done, frankly speaking.
Because
The
why I
reason for
asked
that is the
why
core
I
of
asked this question is that long back when I was doing my PhD, a speaker was invited from hetero drugs, and he has asked this question, but he did not answer it. He was telling that you have to brainstorm yourself and you come back. So that's the reason why, and I got an opportunity here, so I put the same question.
So the thing is, now I can give my perspective, but I don't think I have done any time measuring the pH of the core. But generally, the core is your buffer, right? The buffer you're using, and that will be the pH. The thing is, I told you about remote loading method, right? So there actually ammonium fast sulfate, right? I mean ammonium sulfate. I think salting in, salting out. They change the buffer of the core. So that's how they load the nanoparticles, no, doxorubicin into the core, right? Obviously, the pH is controlled by how you are loading, like what you are loading into the core, right, and how you are loading. I don't know. I haven't come across a way to measure the pH of the core itself. It's generally an assumption based on the method you are using on the buffer. Like say, for example, salting in, salting out, remote loading method of doxorubicin into liposomes, blank liposomes. It will be dependent on the buffer you are using when you are doing the loading, right? So, and then it will undergo dialysis and all the purification steps, right? Irrespective of purification steps, the loading happened when you are loading with the doxorubicin, right? That is with the buffer and the solutions you are using. Obviously, the pH will be of the buffer you are using, right? I don't know. I don't have a clear-cut answer, frankly speaking. Yeah.
Thank you very much, sir.
Greetings for the
yeah. Please go on. Please go on.
Sir, I have a small doubt regarding that formulation, sir. Actually, you have explained about that liposomes and various lipid-based formulations, sir. Thank you so much, sir. This was the very informative session. I have a small doubt regarding that. What is the exact difference between the self-nanoemulsifying drug delivery systems, liposomes, along with that solid lipid nanoparticles, sir? In all the formulations we are using to reduce the particle size. After reducing the particle size, we are increasing the surface area. Thereby, we can enhance the solubility. Thereby, it will impact on the bioavailability. What is the exact difference between the three formulations, sir?
Yeah. So can you see the slide I'm showing now? This is from the precision.
Yes, sir. Yes, sir. I can see, sir.
So this is a lipid nanoparticle, right? This is a liposome, and this is an emulsion, right? This emulsion, which is having a solid core, oil core, and then a surfactant around it, and then a water, continuous phase of water around it, right? This emulsion can be either made by the two methods I showed you, like this method or this method, right? That is an emulsion. Okay, this can be made by self-assembly. This can be made by high pressure homogenization method, right? Of course, the formulations are different. In case of self-assembling nano-emulsion, it can only be done with non-ionic surfactants. You cannot do with any other surfactants. Okay, and because and coming to the lipid nanoparticles and liposomes, the structural difference, like in this slide here, one second, clearly explains this is a liposome, right? This liposome is having a bilayer, which is similar to your cell, any biological cell membrane, right? And this is a cytoplasm, right? And that liposome is like that. And in case of LNP, it doesn't have a water core like here. It will have a solid core comparatively, right? And the only difference between these two, as I explained during my presentation, is in case of liposomes, I'll go to this slide to explain that, sorry. One second. So this slide, right? So you can see the carrier materials difference, right? You're asking the difference between, this is a better slide to explain. The carrier materials in your nucleic acid based liposomes is ionizable lipid, phospholipids. I'm not talking about the payload here. Cholesterol, PEG lipids. But in case of liposomes, you will not have this ionizable lipid. It will just have a cationic phospholipid or anionic phospholipid, or just a neutral phospholipid and a cholesterol and a PEG lipid. And it forms a bilayer membrane, and this forms a structure like this. You can see the structure difference, right? It is a lipid nanoparticle. It doesn't have a water core, right? And in case of emulsion, you just have oil, surfactant, and water, and obviously your payload, right? Payload can be, you know, small molecule and stuff, right? This is the difference. Did I answer your question, or?
Yes, sir. Actually, I had completed my review on self-nanoemulsifying drug delivery system, sir. One of the reviewer asked that question, sir. That's what I have asked here.
Okay. So the thing is, you know, as one of your colleague actually, when introduced, there are different kinds of particles, right? So they label them differently.
Yes.
My lab back then, it labeled it as SANE, self-assembling nanoemulsion. But a lot of people, so all these technologies used, if it is heat-based method, they use phase inversion, irrespective of what methodology. Of course, they get IP, intellectual property, based on the ingredients they use, but because they might have some exclusive ingredients, they might make their own non-ionic surfactants and stuff. But generally, phase inversion temperature is there. That concept is there even during my PhD days. And when this was invented in our lab during IP filing process, they thought this is something innovative, and they filed an IP. Then I actually sent them a book, which I was reading, which already published around phase inversion temperature, right? So I think now at this stage, it is very complex. We need to talk about the ingredients you're using, right? And, you know, in this, I will be very frank, I used an oil, which can be rice bran, soybean, anything, and then a soybean surfactant, which is tween 80, or a solitol HS15 back then, which is non-ionic surfactant, and then water. That's it. So I don't know what you are using. Depending on that, we need to talk about it. So did I answer you?
Just I'm in initial stage only, sir. I wanted to do that research. Yes, sir. Yes, sir. Yes, sir. Thank you so much, sir. Sir, I have one more question, sir. So just I have started my research work on self-nanoemulsifying drug delivery system, sir. Still, I am doing the literature review only. Can you give me suggestions regarding that, sir?
Yeah, you can contact me at my personal email, srikakumanu@gmail.com. At Gmail, Sri Kakumanu, my first three letters of my first name and my last name @gmail.com, or my school affiliation, my first name _ last name @uml.edu. Shall I repeat?
I will.
Sir, please, sir.
Yeah, my first name _ last name @uml.edu.
uml.edu.
Yes.
Sir, can you please provide in chat box, sir? That is the better.
It is given already in the chat box.
Okay. Okay.
Yeah. Thank you so much, sir.
No.
Sir, I have one more doubt.
Yeah, sure.
Actually, I have worked with siRNA. You are loading siRNA into liposome. So can we do nucleofector to load siRNA into the liposomes?
Can you please explain? For example, for sure, siRNA can be encapsulated. That is the centerpiece for the success for a lipid nanoparticle. Onpattro, the particeran from Alnylam, encapsulated siRNA into a lipid nanoparticle. Generally, as I told you, like all these are lipid particles. They tend to name them differently depending on how they structurally assemble, right? And obviously, yes, sure, you can encapsulate an siRNA into a lipid nanoparticle, and that is already proven. And that's the mother of everything, actually.
But I'm asking that can we use nucleofector to transfer sRNA into liposomes?
I don't know. I have to, you can write that email to me. I have to find it. I don't think I have an answer right.
Because I have done siRNA. I have transferred siRNA into the cells, normal cells, with the help of nucleofector.
Yeah. Yeah, that
Uh,
is already proven, right? Like say, nucleofactors or lipofectamine and all those things. These are proven, but they are having toxicity and they cannot be taken into clinic.
oh,
Right? They cannot be scaled up. That's the reason you are having lipid nanoparticles, and that's the market. Yes. Yeah, you're right. It can be done. But what you're asking is, do you want to load a siRNA using a nucleofactor into a lipid nanoparticle or liposome?
right.
I think, I think when you have an opportunity to use nucleofactor anyway, use a cationic lipid.
Yes.
And instead of that, why don't you use a cationic lipid in your liposome or lipid nanoparticle? It will automatically simultaneously encapsulate your siRNA.
Because during manufacturing, there may be a loss of this is a small interference RNA may lose its structure or integrity.
No, they have figured out methods. That's the gist of my presentation. They have figured out methods to encapsulate almost 98%. You will not lose it. Yeah.
Thank you.
So as the time we have passed already time, so we will stop the question and answer session now, and I request Dr. P. Saujanya Madam to give the vote of thanks.
Thank you, sir. Thank you for giving me this opportunity. An ignited mind is the most powerful weapon on the earth, above the earth, and under the earth, and this is got through education. Education is the most profitable investment for the near future. With this positive quote, I wish you all good evening. On behalf of Vignan Pharmacy Institutions, I, Dr. P. Saujanya, Professor and Head, Department of Pharmaceutical Biotechnology, Vignan Pharmacy College, deem it a great honor to propose vote of thanks on this memorable occasion of organizing collaborative webinar on overcoming challenges in the formulation of lipid-based nanoparticles and their present applications. First and foremost, I give glory to the Almighty God for making today's webinar successful. Our deepest sense of gratitude to Honorable Chairman Dr. Lau Rattai Garu, Vice Chairman Sri Lau Sri Krishna Devarayulu Garu, Chancellor Dr. K. Ramurthy Naidu Garu, Vice Chancellor Dr. M.Y.S. Prasad Garu, Registrar Commodore Dr. M.S. Raghunathan Garu for providing us this opportunity in organizing the webinar. We are grateful to Speaker of the Day, Dr. Srikanth Kakumanu Garu, for sharing his valuable knowledge and excellent presentation. It's very useful information, sir. Thank you so much. Our heartfelt thanks to Head of the Institutions of Vignan Pharmacy Institutes, Dr. C.H. Jitendra Garu, Dr. P. Srinivas Babu Garu, Dr. Y. Srinivas Rao Garu, Dr. M. Srinivas Murthy Garu for their continuous encouragement and guidance in bringing out this webinar to the participants. My special thanks to the organizing committee members, Dr. K. Purnanagasri, Dr. P. Raghuveer Verma, Mr. B. Prabhakar Reddy for their valuable contribution in organizing the webinar. I thank all the teaching and non-teaching staff members of Vignan Pharmacy Institutions and all good hearts who worked behind the screen for their support in organizing the webinar. Finally, I thank the wonderful participants who have turned up in such great numbers. Thank you so much for your cooperation. Once again, thank you all. Happy learning. Have a good day.
Thank you.
Thank you very much. All the participants, I hope the feedback link is shared in the chat box. You can fill it. Thank you for your participation, and I thank all the heads of the institutions once again. Thank you, sir. Srinivas garu?